Hello, word nerds. How are you doing today? This episode is another fantastic episode. I'm going to say that right now, even though it hasn't happened yet, because we have another guest, and this is the beginning of a few guest episodes coming up. Today we have Camyla Rocha, and I want to know why are you here today, Camyla? Please tell us.
Well, I'm here because you reached out on Facebook asking for are any DNA nerds out there, and my husband volunteered me, and here I am.
Yeah, he sure did volunteer you. Well, you accepted. I wouldn't have forced you to do
this.
Oh,
no, for sure not. I had him message you in private and say, hey, I'm available.
Yeah, that was great. And I should mention that your husband is one of my oldest friends in the world, Jonah, who has made one of my theme songs. I have two theme songs, and he's made one of them. I alternate back and forth each episode. I couldn't tell you now if your episode is going to have his theme song or not. There's a 50-50 chance. Hopefully it is. I couldn't tell you, but yeah. So he has made that wonderful theme song, and of course he's been on a couple episodes in the past, and so now we've got you joining the Dictionary Club, and then maybe someday when your son is old enough, we'll get him on here as well.
Sure.
I'm going to be doing this thing well until he's an adult, so there's a good chance.
Jonah said it will be a few years until you're done with this project.
Just a few. I'm thinking at least 20.
Okay.
Yeah. I'm making my life really hard. So why did he volunteer you? Why do you know more about DNA than I do?
I currently have a bachelor's degree in biology, which I found an appreciation for molecular biology during my studies. So I've been focusing a lot on the molecular level of just knowing about biological stuff. So DNA is very much a big part of that. And I am currently also working on my master's. We are trying to engineer a bacteria to be able to digest cellulose a little better. So that also involves a lot of knowing about genes and where to put a gene into a genome and stuff like that.
Oh man, that stuff is so fascinating, and that's where my brain just sort of shuts off. How do people even do this? How do they think about this? How do they know what to do? It's just I'm flabbergasted at that kind of stuff.
Yeah, it's really amazing. Right now, I've been writing more than doing actual lab work, which is a bit boring, but it's also a very much needed aspect of the work that you need to get done.
Yeah. A good balance would be nice.
Yeah.
That is incredibly impressive that you are. So is your master's, when it's all said and done, it will be a master's in molecular biology, or is there a more specific name for what you're doing?
It'll be a master's in biotechnology.
Oh,
yeah.
Even fancier sounding than molecular biology. Biotechnology, oh my God. Well, if you have any additional information about sort of where you're working, and you're still in school obviously, but what you're doing, if you have links that you think people might find interesting, send them over. I'll put them in the show notes if they want to go check out more information of just also what is biotechnology.
Sure, I can certainly send you a few links. I belong to the American Biotechnology Guild, or society is what they call it. So I can certainly shoot you a link for that.
That's awesome. I only know the basics of DNA from just junior high school, that sort of thing, which I think is absolutely fascinating, and so I really wanted to do this topic justice. And most people probably have more knowledge of DNA than I do, but you definitely have more than I do. And it was a very easy find of oh, I know this person. Let's get her on the show. we made this happen real fast. So let's talk about the words. We have a very odd little grouping here before we get to the word DNA, because it's a bunch of acronyms really, or abbreviations. So I don't think we're going to have a lot to say about these, so we'll just get through them quickly. The first one is DLS, all caps. And I did send you a photo. I don't know if you have that, if you want to follow along.
I am looking at it right now.
Awesome.
Yeah.
No, not necessary at all. Some people like to have that. So DLS is an abbreviation for Doctor of Library Science. I actually know a couple of people who went to school for library science, but I don't think they got their doctorate, so you couldn't call them a doctor. This is a different kind of science than what you do.
I assume so. I don't know anybody who has a library science degree, but I imagine it has something to do with knowing where to keep books in a library organized, and getting through all the articles and being able to organize them in a library.
See, that's as much as I would think. What's amazing is, like, my thought is, what's involved with library science? And I think there's so much more than I could even imagine. Like, I'm sure there's legal issues that you have to deal with, like things that I couldn't even come up with. But yeah, first and foremost, it's like the Dewey Decimal System. How do you organize everything? That's like step number one, probably. Okay, so I don't know if I or Jonah had mentioned this to you, but after each word, I like to do a little sound effect. Just, as a palate cleanser, if you will.
Okay.
So what's sort of like the first sound that you can think of? And we'll make this sound with our mouths. I guess we don't have to make it with our mouths. We could do our hands or something.
The first sound that comes to my mind is just a sound that Harry does a lot. And whenever he stops to observe something, and then he wants to comment on it, he goes...
Oh, that's good. And that's unique, too. There's a story behind it. I like that.
Yeah.
So you just made the sound effect. That's great. So we are now moving on to our next word, which is DM. This is all lowercase, and it is an abbreviation for decimeter, which is, it's just a tenth of a meter. That's all I got for that.
Yep.
I like this. I would have thought of this sound effect. It's fantastic. The next word is DM again. This is all caps, and it is an abbreviation for Deutschmark, which I don't think they use this anymore, but this is the money that was used in Germany. I think I do remember reading that, but now I think they're on the euro. So the Deutschmark, that's it. We're done with it.
Interesting.
Have you ever been to Germany?
No, I do want to. I've tried to learn German on Duolingo several times, and it never took off.
Yeah, it's one of those things that's like so easy to stop and so hard to start up again.
Yes.
Yeah.
Yes, very much.
It is your turn to make the sound effect.
The same one?
And you feel free to change it up in any way, or just do the same thing, whatever you want.
Let's go with...
You can do it's the same sound, but like if you want to do it in a different way. Sometimes I would go like t t t t t or t t t t t t. I just have fun with the same sound effect.
Okay. Da, 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 da.
There you go. That was definitely different. No pressure on changing it. If you want to be consistent, that's fine too.
It's just I'm going through Harry's sounds. Honestly, it's everything that he does. I just repeat to him. It's gotta be pretty amazing to just sort of watch a child grow in that way, and like just watch the world through their eyes. Oh yes, it is. He's just taking his first steps. Like right now, he took 5 steps using a little walker. It is really, really, really amazing. And he picks up stuff so fast. He'll just observe. He knows how to go through the apps page of my phone just from watching me do it. It's amazing.
Yeah. Oh, they're like sponges. It's amazing. I really do hope to get him on the show someday, actually. That would be awesome.
Maybe when you're in the Z's, we'll get him on.
It'll be before that, for sure. All right. The next word is DMA, all caps, abbreviation for Doctor of Musical Arts. And I guess you can get a doctorate in musical arts. Jonah probably could have done that.
Yes, I am actually pushing him to go for it.
Really?
We heard about a program that's been going on in Vanderbilt that you can get a slow master's.
Okay.
Like take just like one class at a semester.
Right.
We're still talking about it, but yes, yes.
That would be very cool. Yeah.
It would.
Amazing that that he has now a connection to a word in this episode that like we wouldn't have even predicted. That's awesome. The next word is D-mark, and I'm sure you could probably guess what this is. It's got a capital D hyphen mark, and you could also say Deutsche Mark. Oh, sorry. It's actually pronounced Deutschmark, but it's spelled D-mark, and it is a noun from 1948, and the synonym is Deutschmark. We get a lot of repetition like this sometimes. Sound effect for you. I can totally visualize a baby doing that. DMD is next. All caps. Abbreviation for Doctor of Dental Medicine. Huh. I've never heard of DMD, but I've definitely heard DDS in relation to a dentist. This is from the New Latin Dentariae Medicinae Doctor. So I get that's the New Latin way to say Doctor of Dental Medicine. I wonder if that even gets used anymore.
No idea.
Nope. You did the last sound. Yeah, so it's my turn. T-T-T-T-T-T. DME is next. All caps. Noun from 1947. An electronic device that informs the pilot of an airplane on its Start over. I'm a dummy. An electronic device that informs the pilot of an airplane of its distance from a particular ground station. That was worded weirdly, so no wonder why I got confused. I'm trying to sort of process this. It's a device that tells the pilot about, is it about another airplane, distance from its particular ground, or it's their airplane and how far away it is to a ground station.
Yes.
That makes sense to you?
Yes.
Okay.
Yes. Like an airport or something.
Right.
Yeah.
Right. And it is DME because it stands for Distance Measuring Equipment. Sounds like a very important piece of equipment to have.
It does. Yes, it does.
In 1947, that's pretty impressive. I wonder, like, how far away do you have to be before it starts measuring? That's all I got for that.
It's right away.
Right. Like, can they pre-program how far away they are first, and then it sort of just deducts the numbers as it goes? Maybe I'll put a link in the show notes so people can read more about this DME.
That does sound interesting.
Sound effect for you.
T.
There we go. Next is D-Min, capital D, capital M, and then I-N. Abbreviation for Doctor of Ministry, which I can only assume is a minister, like in a church or something.
That's what I would assume as well.
Yeah. DMSO is next. All caps. Noun from 1964. Ooh, and if you are a patron, you get to see potentially see a beautiful cat now in the way, which is awesome. Sometimes when I do these records at home, which has been a while, my cat Audrey will pop up next to me, which I always love. So sorry, you're not a patron, you don't get to see the cat.
Yeah, Benny takes any chance that Harry's not on me to do this. Yeah,
for sure. Okay, so DMSO, noun from 1964. The synonym is dimethyl sulfoxide. And is that something that you have any knowledge about, being in the sort of molecular biology world? I don't know what it is.
You know, it's a chemical that I know of. I think chemists use this a lot. It's used to help reduce inflammation.
I am going back a number of pages because I probably talked about this. Here we go. Dimethyl sulfoxide, a compound obtained as a byproduct in wood pulp manufacture and used as a solvent and in medicine as an anti-inflammatory agent. See, you know your stuff. I never would have known that without reading it in the dictionary.
I knew it was a chemical, and I kind of had an idea that it was used for inflammation, but I had to look it up just to make sure.
And have you usually heard it as DMSO or? Yeah. Okay, gotcha. Next is DMT. This is a whole different type of chemical. It is a noun from circa 1966, and the synonym is dimethyltryptamine, which I believe I did talk about. Now, I think it mentioned it when I read it. Are you familiar with this? Have you ever heard of this one?
I am trying to look this up just to make sure, but I think I may have a tattoo of this molecule.
Oh, really?
It's a psychedelic drug. Where's my tattoos folder? So I got a tattoo because I have very vivid dreams, and that's something that I've always had. And I wanted something that I could that was interesting enough that I could put a little bit of Harry Potter and a little bit of Doctor Who and a little bit of Supernatural. So it's a huge molecule that has like a few different nerdy geek stuff around it. But because of my dreams and all the vivid dreams that I've had, and I looked up what is the substance in your brain that makes you dream. And I think my solution was a DMT molecule that apparently that is the last molecule that your brain secretes right before you die.
Yeah,
and
I've heard
that's
that too.
like the thing that you think of as you're drifting away.
Yeah, I've heard a little bit about this. A friend of mine lent me a book about it, which I never got around to reading. I wonder if I still have it actually. But yeah, so what the dictionary here says, because I just went back, a naturally occurring or easily synthesized hallucinogenic drug that is chemically similar to but shorter acting than psilocybin. So yeah, it's a fascinating thing that people do take recreationally, but it is also just occurring in humans and other animals that, yeah, I think I've heard dreams. It has it as an effect, dreams, and then of course before you die. So yeah, it's a fascinating thing. I will say this, I have never taken it recreationally. I've never ingested this in any way. I am curious though.
I am too. And you know what? I just found the folder that says for sure which molecule it is, and it is DMT. I wasn't sure if it was DMT or DNT, but it is. This is it?
Yeah. And I love the fact that you added a bunch of nerdy elements to your tattoo. That's fantastic. What would you say was in there? Harry Potter and what else?
It's Harry Potter, Supernatural, and Doctor Who.
Oh, gotcha.
So I have a TARDIS.
Yeah.
Like embedded into the molecule, and then a little bit of Hogwarts Tower that has a little flag, and the Supernatural Impala car all the way to the bottom.
That's cool. My niece is a big Harry Potter and Doctor Who fan. I don't think she's familiar with Supernatural. I think that might be a little more adult for her 13-year-old self at this point.
That's when I started getting into it.
Okay. All right. I'm going to definitely recommend that to her then. I think I watched the first couple episodes and I just didn't have the time to keep on going, but I know it's like a huge cultural phenomenon.
It is. I will say it's a huge time investment because there are like 16 seasons out.
That's insane. They're not. Are they still making those?
No, that was the last season.
Good.
Yeah.
That's a lot of seasons. All right. Well, I may need to get a picture of your tattoo so I can show my niece at some point.
Sure.
Okay. Moving on. I think it's your sound effect time.
Oh, yeah. Let's go with...
Nice. We got a little ASMR there. This one is DMV, all caps, abbreviation for Department of Motor Vehicles, everybody's favorite place to go. I'm sure I could talk about the DMV for a while, but I won't because I don't want to bring up terrible memories. I assume that you've had plenty of experiences with that place.
I've had a couple, yeah. None too bad, except for when I had to take my driving test.
Your first driving test?
Yeah.
Man.
Not the actual driving, the written portion.
Oh, okay.
The test, yeah. It was annoying, but
I was convinced that I did not pass my driving test. I think I messed up a couple of things, and the guy didn't seem very impressed. And when we got back to the DMV, I was ready for him to say you didn't pass, and I did somehow. I guess their standards are really low.
That would explain a lot.
Yes, it would explain a lot from all the terrible drivers I see out there. Oh my God. Oh, the DMV. Okay. I have to mention Patty and Selma from The Simpsons because they work at the DMV and they are fantastic characters.
Yeah.
Next is DMZ, all caps, abbreviation for Demilitarized Zone, a place where there is no military anymore. Perfect, thank you. DN is next. This is an abbreviation for down, so we just took out half the letters to make the word DN from down. That's it. Here is our big word. Not only is it our big word because of our guest today, but also because it is literally taking up the most space in this section. So we have DNA, all caps, noun from 1944, and I'll say the etymology first. It stands for deoxyribonucleic acid. So the D from deoxyribo, the N from nucleic, and then the A from acid. It is, it's a long definition, any of various nucleic acids that are usually the molecular basis of heredity, are constructed of a double helix held together by hydrogen bonds between purine and pyrimidine bases, which project inward from two chains containing alternate links of deoxyribose and phosphate, and that in eukaryotes are localized chiefly in cell nuclei. And it says to compare to recombinant DNA. Just real quick, recombinant DNA, can you just briefly tell us how that is similar or different?
Recombinant DNA is just DNA that has been, so you have one leg of the chromosome is from your mom and the other one is from your dad. Recombinant is when they kind of cross over and you get a little bit of each part.
Okay, interesting. Yeah. I might have to have you back when we get to the R's to talk about recombinant DNA when Harry's in college. So there is a picture. It might be a little complicated to describe, but I guess first of all, just what additional information can you give us that sort of is left out? Like, what are some other important things to know about DNA?
I would say that there are 4 nucleic acids, and one is adenine that pairs with thymine, and cytosine that pairs with guanine. And so DNA is a double helix, and each one of those nucleotides have to be attached together. I'm sorry.
That's the cat walking on the piano. This is perfect.
So for them to be attached together, think of the DNA as a ladder, and then the A pairs with the T, the C pairs with the G. But for the DNA to be compacted into the nuclei of a cell, it has to be twisted and twisted and twisted, and we call that supercoil. So that's the supercoiling of the DNA, and if you unpack it, it becomes like this I think it's like 2 meters long.
It's something crazy.
It's a very tall person. It's like over 6 feet.
Definitely taller than me.
Yeah. I would say that that's the most important thing. And then you, of course, you can delve into it a little more. DNA can be wrapped around these big proteins, and then if you want to access the DNA, then enzymes come into it that are able to unravel the DNA so you can access the exact part that you need to access.
Okay. So most of what you said is kind of what I've already known. You know, it's the double helix, it's twisted. That's what we see here in the picture. And I knew that there was the ATGC, but I couldn't remember which ones pair with which. So the A and the T are always paired together.
Yes.
And the G and the C are always paired together.
Correct.
And so to give a little bit more of a visual description of kind of what you had said, if you take a ladder and the one half you said is from your mom and the other half is from your dad, is that kind of how it works, or am I simplifying it too much?
No, that's an oversimplification. That's a chromosome. So the DNA will be super coiled into a chromosome. That's what a chromosome is.
Yes. So that was actually going to be one of my other questions, is where is this DNA stored?
It's stored into the nucleus of the cell. And for you to be able to access that DNA, you have to go into the nucleus of the cell. There are some DNA that are stored into the mitochondria too, but that's a different kind.
Okay. And every single cell, this is a question for you. Every single cell in your body will have the DNA in the nucleus?
Yes.
That's a lot of cells.
Yes, that is correct.
And that is a lot of copies of your DNA that's like 6 feet or more for each one.
That is correct, yes. Not all of the DNA is active. Actually, like over 99% of the DNA we don't really know much about. We have we share 99% of the DNA. Only 1% of you and me are different, basically. And it's not all of the DNA is active all the time. There's a lot of genes that are just like dormant. And the human genome has been mapped completely. But like I said, we don't know everything.
Right. There's still so much to figure out.
Yeah.
So I had that one thing wrong about one half coming from your mom and one half coming from your dad. So we'll ignore that part for now. But basically, so it's the ladder, and there's the two vertical pieces, and then the rungs that go across horizontally. Those are what would be made up of the A and the T. So one half would be one half of the rung that you step on or grab onto would be the A. The other half would be the T. The next rung up could be AT or TA or GC or CG. It's like any combination of those 4 letters with those 2 pairings. And that's what I think is kind of amazing, is that it's only 4 letters. It's only 4 things, only that can be combined in so few ways. But because there's how many ever millions of them throughout the whole DNA, it creates this incredibly unique thing that makes up you.
Yes, that is correct. I should have worn my protein shirt. I have a shirt that has all the nucleotides that translate into different proteins and stuff.
Oh, that's very cool.
But yeah, that is correct.
So I will try to quickly describe this very complicated image that's in this book. Obviously, I'm going to, put a link in the show notes for more stuff about DNA, and probably post some pictures on social media as well. But so there's two things here of DNA, and feel free to jump in at any point with if I say something wrong or you just want to add additional information. So the A image here, DNA A, it's called the molecular model, And the B image is called the double helix. So the double helix is really just showing the basic structure of the double helix, of these sort of two strands that are twisted kind of around each other. And then it shows these sort of dotted lines where kind of those rungs would be of the ATGC. But back to the molecular model, this is the DNA with all of these sort of bubble sphere molecules on it, so it makes it look very, well, bubbly, basically. How accurate do you think that image is of actual DNA?
That is actually pretty accurate.
Okay.
It's got the backbone, the sugar phosphate backbone there, made up by so each circle is patterned differently, and each of those circles stand for a different atom.
Yeah.
Or a different aspect of the DNA double helix. So if you look at it, number 5, it's the phosphorus that hangs attached to the sugar phosphate, to the sugar base, nitrogenous base. That is pretty accurate, actually. I'm impressed.
So yeah, we have these 5 molecules, or these 5 numbers. There's a little key that shows you the circle size is slightly different, and then they have different shadings. Like 1 is completely open, number 2 is filled with a lot of little dots, number 3 is vertical lines, number 4 is just a gray tone, and then number 5 is completely black. So I'm not going to tell you where all of these are exactly, because that would be way too complicated. But basically, number 1 is hydrogen. That's the open circle, or the white circle. Number 2 is oxygen. Number 3 is carbon in the helical phosphate ester chains. Is it helical or helical? It's probably helical. Helical. Number 4 is the carbon and nitrogen in the cross-linked purine and pyrimidine bases. And then, like you said, number 5 is the phosphorus. So,
I do have a question, because I am English is my second language, and I've noticed that you're saying purine and pyrimidine, and I've always said it purine and pyrimidine, and I'm wondering if that is an accent thing.
I'm not sure. You would be way more correct than me, because you actually study this stuff.
I'm just very I have very spotty memory when it comes to specific things like this. I don't remember my teachers saying one or the other. So I am genuinely wondering if I've been saying this wrong the whole time.
These are not words that are common in my my everyday language, so I'm kind of only basing on what my brain wants to say, and I have heard them before, but yeah, I don't know if I'm supposed to say purine or purine. So you said you were thinking purine.
That's
purine
what I thought.
and pyrimidine.
Pyrimidine, yeah.
That makes more sense. But I think because of the INE at the end, that's my brain wants to go ein, but I really have no clue.
Google says it's een.
There you go. I trust you and Google together.
I trust Google.
Yeah. What's interesting, though, is that there have been a lot of words that I've come across doing this podcast that I kind of always thought that they were one way to be pronounced, but it does say that, these 2 or 3 different ways are perfectly fine as well. So I'm actually trying to find this word, purine, to see what does it say. Is it only going to say purine, or is it going to say purine as well? Probably not. Let's see real quick. Purine. Yep, purine. That's the only one in there. So Spencer was wrong yet again. I'm wrong all the time.
So am I.
Yeah, it's such a big thing to talk about. I'm trying to think. There were some other questions that I had for you before, and I've completely forgotten them. let's see. So it's tiny, it's coiled up, it's in all of our cells. We share so much of it. I mean, same with like chimpanzees and bonobos. We share so much of our DNA. There's such a tiny little difference. I think that the fact that like it's a relatively simple structure. I mean, this picture doesn't look simple, but like in general, it's a relatively simple structure of these things. I don't even know what to call them. That just create that, like this is the instructions that tell your body how to create. It tells your cells how to divide in certain ways to make up your physical structure.
Yes.
And I know that there are mutations that happen. Now, will those be in the DNA or the chromosome or both? How does that work?
So every time we go through a cell cycle, like meiosis, That's
when the cell is splitting.
Mm-hmm. There are checkpoints that your body goes through to check that there aren't any errors. And if there are errors, then the cell goes through apoptosis, which is cell death. So that cell doesn't get divided. And there are other mechanisms also that the body can self-regulate. That's actually basically how cancer happens when your body loses the ability of surpass those checkpoints, and then a mutated cell just keeps on dividing and dividing and dividing. It grows into something crazy.
The fact that our body has this checkpoint system in the first place is
it's
incredible.
Several of them. I think there are at least 3, if I'm not mistaken, 3 checkpoints. And
then that it's not only does that happen, but it can also tell the cell. Like, does this happen in the cell itself? Does the cell check, or is it something else checking the cell?
So there are, I would call looking at the meiotic process, I would call it as a cell thing, but there are several tiny different responses that tell you which gene is functioning, which is not. So it's a lot of tiny proteins involved into and signal cascades involved into getting that information to see if the cell is ready for division or not. So you can look at it at a I'm not going to say macroscopic, but like at a zoomed out level, which I think would be the easiest to explain to people. Mm-hmm. Going into like the actual molecular level and looking into the tiny bits and pieces and the cascade signals and the little peptide other signal cascades would be out of my depth to explain. Yeah. But it basically, for those checkpoints to happen, the cell is checking constantly while copying genetic information to make sure that everything is where it's supposed to be. And if one of those signals are not there, or if there is something that is there that is not supposed to be, the cell is usually pretty good at catching that. The cell is pretty good at catching mutations at the nucleotide level, because after they copy the whole thing, it goes through a proofreading process, and then it's really wild.
It is.
And then after that proofreading process, there are some mutations that can slip by, but some mutations are too detrimental. So if there is like if there is a mutation that makes translation stop too early, then that gene is probably not going to be completely copied. And that means that if it's a very, very important gene then that the cell needs to survive, that cell is just not going to complete the cycle.
And then it just the body was like, nope, sorry, you can't divide. We don't want you anymore.
Yes.
If it's working properly.
Yes. It goes into apoptosis, and sometimes parts and bits and pieces of that cell can be recycled into your body, or at least for energy, but sometimes it doesn't.
Man, the body is like it's just astounding what it does, what it can do. This is like we don't have any consciousness of what's going on. Like, we're not aware of this. It just happens, and man, it just blows my mind. It really does.
Yeah. That's why I went into biology.
Yeah, it's fascinating. So, like, what? I'm trying to think of like what are you studying? Like, what's like one thing that you're super interested in, or that you're studying now, or that you've learned that you find just incredibly fascinating? I don't even know what my question is.
Well, what I'm doing right now is we're trying to use a specific bacteria to, I'm going to say the word absorb, but so we're trying to put a specific gene into this bacteria so we can digest cellulose better. And if we can make that happen, then perhaps instead of using the fungi that has that gene that takes a specific set of conditions to grow in a lab, it takes longer to grow fungi in a lab. Bacteria, you can have like a day output. Like you feed the bacteria, next day they are grown, and if they are secreting the enzymes and the genes, if they're expressing the genes that you're interested in, that's good. So my project is making sure that bacteria can take up this good cellulase gene, and hopefully I will be able to make that digest cellulose. And in an industry level, it could be used for ethanol fermentation, so like to make biofuels and stuff like that. If we can get that to digest paper or wood cellulose, then that's a good thing.
Yeah.
But my project is just like the very beginning of that.
Right. Yeah, I mean, that could potentially lead to a lot more renewable fuel sources, because we're, obviously our whole oil situation is messed up.
Right. So my advisor is very much interested in renewable energy sources within a biotechnological approach. So what tools can we develop that would make that reality more attainable, basically? So the tool that we're working on right now is this bacteria, the specific bacteria that I'm working with.
Are there other students trying other things?
So yes, he works with yeast mainly. This is Dr. Robertson at MTSU. He has several papers that he published that he was able to build tools that would make biotechnology better. He designed a bioluminescence reporter years back with another student for circadian, like rhythm, yeast day night cycles. It's been a while since I've read that article of his, so I don't know the specifics. And then there is another student who is working on his own project, has to do with tobacco. And me and TJ, which is a PhD student in his lab, are the only ones working with this specific bacteria. And but his project is a little different than mine. He's focusing on polymer chains, breaking down polymer chains.
Gotcha.
Yeah.
Man, yeah, this stuff is so fascinating, and it's been really interesting to see sort of where we've come in the last 30 years and where we're going, especially because it pertains to, biofuel and things like that.
Yeah, it's interesting. I before getting my bachelor's in biology, I was like very, very interested in cloning, because all that CRISPR talk was everywhere for a couple of years back, and everybody was all about CRISPR. And then in taking all the classes that I've taken, I've learned that there are easier traditional cloning techniques, which is mainly what I use for my project. We don't really do CRISPR in the lab. We just do stuff that's been out there for over 50, 40 years. That's like old school science. Science is what Dr. Robertson calls it.
Old school, yeah.
It works. It still works. And hoping my project will be successful.
I hope so too.
Yeah.
And that's how long do you expect that to? When do you think you'll find out?
It depends on the experiments that we're trying to set up. So far, it hasn't been great. I'm trying to write up my proposal in the next month so I can defend it. So it's mostly going to be background information. Why do I think this is important? And telling them what it is that I want to do to make it happen. But I intend to graduate after the fall, so like in December. So hopefully by then, in a year,
well, maybe less.
Yeah, I'll have some data to show that I can actually write my thesis on.
That's very cool. And then you're out in the world, and you'll find another project to work on.
Yeah. Hopefully, well-paying job.
Yes, that's always the goal. Well-paying, but also fun and interesting.
Yes, for sure.
Yeah.
I don't want to just write stuff in an office.
You want to do actual experiments.
Yes, correct.
Yeah. I feel like my brain is very full with all of this information. I find it super interesting. And, if anybody listening has, even the smallest amount of interest in DNA, molecules, all that stuff, go learn that stuff. Go to school to learn that. And yeah.
I highly suggest basic biology courses.
Start there.
Just to get the main idea first and then move on. Genetics was what hooked me into the whole thing. I never studied it, but I find it fascinating. All right. Would you like to do one more sound effect, please? Yes. Let's go with tee tee.
Does he actually make that sound too, or
No.
just nice?
No, I'm
okay.
just trying to change it up.
Perfect. This is our last word. It is DNA fingerprinting. 2 words, noun from 1984. So DNA was coined in 1944. This is 40 years later, and it is a technique used especially for identification as for forensic purposes by extracting and identifying the base pair pattern in an individual's DNA. It is also called DNA typing, and a DNA fingerprint, that is also a noun. So the fingerprinting and the fingerprint. We, every once in a while, my wife and I will watch some, true crime shows or anything related to that. And, for the last 30 or 40 years, they have been able to talk about DNA and how that's played such a huge role in forensics. Could you just give kind of a quick, more of a description than what they gave here, just describing sort of how this works and like what's involved in this?
I don't know much about how their process works because I haven't really taken any forensic level classes. That's like a different department.
Yeah.
But I do know it involves, this is a wild guess. Maybe they try to sequence the DNA. And then they try, because I know it involves a gel, running it through a gel, getting specific bands on the gel, and then comparing it to like whatever sample they have to the suspect and seeing if those bands match around the same areas on the gel.
I've seen those pictures of those bands and I never really fully understand like, what is that? But I guess it's a visual representation in some way of a person's DNA.
Right. And it does involve a letter. Like it's what we call a letter, which is a known sequence of DNA cuts. So there will be 100 base pairs right at the top, 200 right at and it goes like there are different sizes letters. Okay. And then that's the first thing that you load onto the gel to compare the sizes of what you get. So, for example, for my project, I told you that I'm trying to incorporate a gene into my bacteria that makes a recombinant DNA. That makes the new DNA a different size than the original one. So if I want to know if my bacteria took up my gene, I cut it with a specific set of enzymes, and then there are programs online that I can throw my sequence through. And then it'll tell me what size to expect. Oh, my gene is 1.2 kilobases. Then I cut my bacteria with those enzymes. I run it through a gel. If I get a band at around 1.2 kilobases, then I know that my experiment was successful. I imagine it's something like this.
Okay. Yeah, I mean, the little bit that I know is, from the even earlier than what you described is, they take a blood sample or a skin sample or a hair sample, something that has our DNA in there, and they'll go through the process that you described or something like that. They'll send it to the lab.
Yeah.
and then the fancy lab people do their fancy lab work.
Right.
And then they're able to sequence the DNA, figure out the specifics of the DNA, what makes that DNA specific to another DNA.
Right. I don't actually know if they sequence it. That's like I said, that was a wild guess. But if you want to know specific bases, then I would say that you would need to sequence it. But maybe you don't need to be that specific. Maybe if you just can get good chunks that match the sample and the suspect. That
get enough information.
Yeah.
Yeah. And then, like you said, compare that to the DNA that they've tested from the suspect or some other people to see if that matches.
I mean, that's what I imagine it is like. I've done theoretical exercises of that on like my first year as an undergrad, which has been 6 years ago. So I don't remember much about those, but I think I may be on the right track.
It sounds right to me. I'm going to say it's right. Yeah. And yeah, I mean, that's had a huge effect on forensics and figuring out who did this thing, who was the murderer, whatever it is. I mean, they've actually gone back decades and solved crimes from way back when.
I've recently also gotten into true crime podcasts.
Yes.
And it's, I listened to one. Can I say the name?
Yeah.
It's just Crime Junkie.
Okay. Oh, yeah, I think I've heard of that.
Yeah. It's amazing storytelling, and they call, like, the recent years, it's the season of justice because of all the DNA stuff. So they are solving a bunch of cold cases from way back when, because now they've collected all these blood samples from, or like any fingerprint samples that they had from years and years ago, from the 80s, from the 70s, and then now they are able to compare all of that information with even relatives, because there's 23andMe now, so you can get some of that information and ancestry. I think.
Are you familiar with the I think it's the Golden State Killer?
Oh, yeah, they did an episode on that.
Yeah.
He was recently caught, right?
He was, yeah. And I think it was from DNA, and I think it was also from 23andMe or Ancestry, one of those. And correct. I mean, this was a story of somebody, God, must have been at least 50 years. I think it was in the 70s, yeah. So like 50 years ago, he was killing all these people in California, or Washington? Golden State, whichever state that is. Washington, I want to say. And it was this cold case for all these years. And then I don't know how much you know about this, but my wife is also big into true crime, so I learned all about this. But there was a woman writing a book about him, trying to figure out who he was, called I'll Be Gone in the Dark. She happened to be the wife of Patton Oswalt, the comedian and actor, she ended up dying before she finished the book. I think because of the stress of writing this book and like everything involved, it just killed her. And some of her co-workers, people helping her out, they ended up finishing the book. Then they turned it into maybe a podcast, but also a 6-part HBO series.
Oh.
Which is about finding him, but also about her story. It's this fascinating series about just all of it and her story. And yeah, it's really incredible. And yeah, they found the guy and he's in jail now. And he's like amazing. 80 or something.
Yeah.
Yeah, it's crazy. Oh, okay. Well, that was a lot of fun talk. I'm so glad that you did this. So the last part that we do is I will quickly reread the words that we had, which are just a bunch of letters, mostly, and you get to pick a word of the episode. I may know what you want, but I could be shocked. So today we had DLS, DM, DM, DMA, Deutschmark, DMD, DME, DMIN, DMSO, DMT, DMV, DMZ, DN, DNA, and DNA fingerprinting.
I'm gonna go with DMT because it reminded me of my tattoo, and I hadn't thought of it in a while.
That is awesome. Yes, I was thinking if she doesn't pick DNA, she's probably gonna pick DMT. That's fantastic. Nice curveball there. Thank you. I appreciate that very much. So DMT is the word of the episode. Maybe I need to get some DMT. See what it's all about. We'll see. I don't think it's legal.
No, I wouldn't do that.
No, probably not. I've heard stories of people talking about it, so I'm like, what's that like? Sounds interesting.
I'll wait until I die and then have the full experience.
Yes, exactly. The full natural experience.
Yes, exactly.
Yeah. Okay, you can totally say no. Would you like to sing a little song about DMT, which could be as simple as just singing the letters DMT, and that's it?
Oh, gosh. Okay. Just me, or can you help me?
Whatever you prefer. There's a bit of a delay, so I don't know how well that would work, but we can try it. Yeah.
I'm just trying to run through the makeup songs that I sing for Harry sometimes and see if that would work.
Yeah. And it could just be as simple as like, DMT, DMT. That's DMT. That's probably what I would do.
Okay.
And if you don't want to, that's fine too.
DMT, DMT, for you and me, it's DMT.
Yeah, that was great. That was perfect. Is that similar to one of the songs that you sing?
No idea.
It was totally new and original.
Yep. Yeah.
Just
for your podcast.
And it will never be said again. You have
I'll probably
to listen
forget
to this.
right after I forget.
Yeah. Well, thank you so much for doing this and having this incredibly nerdy conversation with me.
Yeah. Thanks for having me.
Anything you want to plug or tell the people?
No,
Just anything.
I'll send you a couple of links for biotechnology resources if you're interested. But yeah.
Sounds good. Well, until next time, this is Spencer dispensing information. Goodbye.
Goodbye.